so well, which we know so well, his work not only in the callosum, but also in development and plasticity of the nervous system. He was a postdoctoral fellow at MIT and Harvard Medical School before joining Harvard faculty, where he has worked for many years studying plasticity and regeneration of CNS connections. In recent years, he's working at the Boston Children's Hospital. His research has focused primarily on regeneration of the optic nerve, studying novel growth factors associated with inflammation, signal transduction, pathways, transcription factors that act as master regulators, and combinatorial therapies to promote optic nerve regeneration. I think that we should abbreviate this very long CV and tell you most of the people here know Dr. Benowitz, and he's currently a professor of neurosurgery and ophthalmology at Harvard Medical School and holds the Neurosurgical Innovation and Research Professorship at Boston Children's Hospital. It's a great pleasure and an honor to have him. Thank you so much for accepting our invitation. Please.
Well, thank you for that lovely introduction, and it's really so nice to see you. The people I've known, some of you for many, many years, and I'm nice to be meeting you, the rest of you, unfortunately just in 2 dimensions. Someday we hope it's in 3 dimensions. So let's see, so should I plan to speak for about 45 minutes or something like that?
That's perfect.
Okay, so let me get my, okay, good. Okay, so I'll be talking about a few topics today, which are very nicely anticipated by Cecilia's introduction. First, a little bit of background in the field. And as Cecilia said, how innate immunity can be harnessed to promote regeneration. Third topic, I'll just speak about very briefly what we've learned about presynaptic influences on optic nerve regeneration, the role that the inhibitory interneurons in the retina play in suppressing regeneration. And then the last point will be our studies of the transcriptional regulators of the regenerative program. So, yeah, there we go. So we're going a little bit deep in history. This is about 5,000 years old. This is an Egyptian papyrus, actually written sometime later. The papyrus is only from 3,500 years ago, but it records the I think someone has the microphone on again. So this records the teachings from way back in the Old Kingdom of Egypt, when the Grand Vizier Imhotep taught about many issues in medicine. And you may know the ancient Egyptians were very advanced in many areas, geometry, medicine. But what this papyrus says, not that I really can read it, but what I'm told it says, it describes many diseases, many disabilities, and what the proper treatment would be. But then goes on to say that if there's injury to the spinal cord, unfortunately, this is an untreatable condition. So that situation remained the same for thousands of years, and we're really only now getting to the position where we might be able to do something about it. So, in the interest of time, we'll skip the intervening 5,000 years and come to Rumoni Cajal, who I'm sure you're all familiar with. Cajal was really the father of modern neuroscience. So after his really epic descriptions of the anatomy of the nervous system, the development of the nervous system, Cajal then turned to the degeneration and regeneration of the nervous system. So this book was originally published, I think, in French in the 1920s. Here's a wonderful picture of Cajal and his students, advanced fellows. And what Cahal points out is what you all know, and that is while regeneration is possible in the peripheral nervous system, it's not at all possible in the central nervous system. And so the model example that he used to illustrate regenerative failure in the central nervous system is the optic nerve of a mature mammal. So he did studies in rabbits, in adult rabbits, but his trainee, Teo, Teo, circled in red here, was a brilliant student of Cahal. Unfortunately, he ran up against the Franco government of Spain, and that was the end of his career. But Teo did a remarkable experiment. He cut the optic nerve, and then what he did was to suture the sciatic nerve, the animal's own sciatic nerve, to the cut end of the optic nerve. And what Teo observed is that some axons that would normally never be able to regenerate in the native environment of the optic nerve itself are in fact able to regenerate into this sciatic nerve graft. So Cahal, having studied this issue for years, remarked how astonishing this was. This represents the first example ever of successful axon growth from neurons of the central nervous system. And of course, the retina is central nervous system. The retina originates in the diencephalon. The axons are sheathed by oligodendrocytes rather than Schwann cells. So it is a model of the central nervous system, not the peripheral. This showed the potential of CNS neurons to grow axons, but of course this was happening in a very artificial environment. And so the next part of the story is the epic work of Aguayo. I'm not sure what that black line is down the middle. But Aguayo then, starting in the 1980s, on through the 1990s, maybe up to 2000 or so, this was really considered some of the most exciting work in all of neurobiology at the time. And those of you who are my age would remember the great excitement that this caused. So Aguayo's group picked up on this model, cut the optic nerve entirely, and fixed the sciatic nerve, the animal's own sciatic nerve, to the cut end of the optic nerve, and then found that some axons originated from retinal ganglion cells here in the back of the eye, were able to send axons to the full length of these peripheral nerve grafts, and they exited here in the superior colliculus, of course a primary normal target of the optic nerve, and here we see some evidence of synapses forming. So this was just amazing, but of course, what still remained impossible was regeneration to the optic nerve itself. And so the next important figure to introduce at this point is Martin Schwab. And Martin has worked he did some of his postdoctoral work here at Harvard, but did most of his life's work in Zurich, Switzerland, and really did tremendously important work. So Schwab picked up on these observations to try to understand why regeneration failed in the central nervous system. And what he showed for the first time is that oligodendrocytes, the glia that ensheath axons in the central nervous system, that oligodendrocytes are strongly suppressive to axon growth. So here in this phase microscopy image over here, what Schwab is showing is here we have a couple of oligodendrocytes sitting off on the left of the center in these culture dishes, and these neurons, peripheral neurons from the superior cerebral ganglion, from the superior cerebral ganglion, are growing nicely in culture, but when, this is too sensitive, but when they encounter the oligodendrocytes and the myelin that these oligodendrocytes make, they are studiously avoiding the myelin. So then, as you may know, Schwab and others went on to identify many of the strong inhibitory molecules associated with myelin, myelin-associated glycoprotein, the novo protein, and experience oligodendrocyte myelin glycoprotein. And at the same time, people like Jerry Silver and James Fawcett and others were showing that the scar that forms at an injury site is also strongly suppressive to axon growth. So at that point, there was a great deal of excitement in trying to promote regeneration by counteracting these inhibitory molecules. But to make the long story short, that lasted maybe 10 years in the literature, there was really very little success from this. And so this is, so I let that slide out. Yeah, okay. So both in the optic nerve and our work and others, and in the spinal cord, people found that counteracting inhibitory influences, which can be done by a number of genetic means, is not sufficient to promote regeneration. So the question then is, why can't the CNS repair itself effectively? Well, one thing we know is that the brain has very little capacity to replace neurons that are damaged. Neurons cannot regenerate axons over long distances. Undamaged neurons have only a limited capacity to form compensatory circuits. So what is it that limits axon regeneration in the CNS? Well, from the work of Jeff Goldberg and others, we know that once neurons mature, the intrinsic capacity for axon growth has declined greatly. Jerry Schneider said that, actually showed that earlier, as Raphael, if you remember. And the levels of trophic factors may not be as abundant as they were during development. The cell extrinsic suppressors associated with myelin and scar that forms, these certainly are important factors, even though counteracting them is insufficient to enable growth to occur. And then in the case of the optic nerve, where injury causes neurons to die, we've pretty much precluded any possibility for growth. And then something that we've learned and others are learning recently is that there are presynaptic influences on neurons that can suppress axon growth. So then the question, I don't think I need to explain to this group, but why is the optic nerve in a suitable model? Well, one answer is it's good enough of a call as well. But a more of a kind of less facetious answer is that the optic nerve is an integral part of the central nervous system. It's the most accessible part of the CNS. The anatomy is very well defined. Vision is important. This is how we get most of our information about the outside world. And also we're learning that mechanisms that have discovered in the system have generally been able to, have generally been applicable to other parts of the CNS. I'll talk a little bit about that at the very end. So now we introduce Martin Berry, who did all of his work in England, mostly at Guy's campus. And Berry was a bit of an iconoclast. So while everyone else in neurobiology studying regeneration was thinking about the inhibitory influences of CNS myelin, Berry was thinking, no, that's not right. I mean, the reason, Berry said, the reason that axons can regenerate when you do this peripheral nerve graft is that the graft that you're putting into the eye, the peripheral nerve graft that you're putting into the eye, has Schwann cells. And maybe the Schwann cells are making trophic factors that activate the retinal ganglion cells to grow their axons into that peripheral nerve graft. So what Berry did was a very atypical experiment. He crushed the optic nerve, but left the distal part of the optic nerve intact. And now what he did, this is a control where he just crushed the optic nerve. These axons are stained for the growth-associated protein GAP43. There's no axon growth distal to the injury site, as expected. But the remarkable finding from Berry's experiments was that when he put peripheral nerve grafts into the back of the eye, he saw something amazing, and that is that many axons grew beyond the injury site. So this was an amazing observation. This was the first time anyone had ever obtained axon growth distal to the injury site. I remember going to a Society for Neuroscience meeting shortly after this paper was published. I was very excited about it. I ran into a number of people in the field who said, no, It's impossible. Everybody knows you can't get regeneration in the optic nerve. But of course, this can't be a mistake because the axons here are stained for the growth-associated protein GAP43, and they would only be immunopositive for GAP43 if they were truly regenerating. So this was really a big deal. I got very excited about this. At that point, I had been studying regeneration using lower vertebrates, trying to understand how they regenerate their axons, looking at changes in gene expression in retinal ganglion cells, proteins that are expressed during regeneration. But at this point, we started doing cell culture studies, putting the retinal ganglion cells from goldfish or mice in culture, and then trying to see what was it that was in the native system, in the endogenous system, that stimulated retinal ganglion cells to grow axons. So we had begun characterizing a small molecule at that point, and we thought, well, let's inject that into the eye and see what happens, see if we can mimic Berry's phenomenon. So this is the first part about the role of innate immunity in promoting regeneration. On my screen, the top right is blocked, but I don't think that's happening for the rest of you, is it? Can you read the caption here?
Yes, it's okay. Oh. We can also see the full slide.
Okay, thanks. Yeah, so we carried out experiments to test the small molecule. So as was already well known, you injure the optic nerve, and at that point, the axons distal to the injury site begin to degenerate, and beginning 5 days or 7 days later, the proximal portions of the axons degenerate as well, the ganglion cells begin to die, and by about a month after this injury, you have maybe 2% or 5% of all the ganglion cells remain alive. So obviously, there's no possibility of recovery, visual recovery. Quite by accident, we made a very fortunate discovery, and that is we injured the optic nerve, we tried to inject the small molecule, but inadvertently we nicked the lens, we injured the lens. This causes a massive inflammatory reaction to take place in the eye. We get an influx of neutrophils first, within the first 12 hours, and then an influx of macrophages follows within the next 12 hours. So say from 1 day on, we have a huge number of macrophages as well. And we did a transcriptional profiling of the retinal ganglion cells when they were stimulated to grow. So, well, first of all, this is sufficient. Just nicking the lens, causing this inflammation, is enough to enable retinal ganglion cells to regenerate their axons. Pretty much to the same extent as what Martin Berry had seen. So they turn on GAP43, SPR1A, ATF3, many other gene changes that are described in, I left those papers out, but maybe they'll appear in a minute, and the axons begin to regenerate. So why is this happening? Why do the axons regenerate? So through extensive biochemistry, biochemical characterization, we found that the invasive neutrophils are expressing very high levels of a protein that we isolated using column chromatography and mass spectrometry, protein called oncomodulin. So here's immunostaining for oncomodulin, and you could see that the neutrophils, which stain heavily for the GR1 protein, surface protein, so these are the neutrophils here. So both the neutrophils and the non-neutrophils, the macrophages, are expressing high levels of oncomodulin, the infiltrative cells. And then, so this is all the work of Yu-Chin Yin. By PCR, we see that levels of oncomodulin in the eye, this is looking at the whole eye, the retina as well as the vitreous, we see very little of oncomodulin in the normal eye, but the levels shoot up as the infiltrative cells enter the eye. By immunostaining, we see that the control eye shows very little of this protein, oncomodulin, but the inflammatory cells secrete high levels of the protein that then cross the inner limiting membrane. And so we now can detect high levels of oncomodulin immunostaining in the ganglion cells, whose cell bodies sit over here in the ganglion cell layer, as well as in the the dendrites of the ganglion cells in the inner plexiform layer, and it looks like it's really staining much of the retina, much of the inner retina. And so when we package oncomodulin together with a cyclic AMP analog, cyclic AMP is required for oncomodulin to work because elevation of cyclic AMP causes the receptor for oncomodulin to translocate to the cell surface of retinal ganglion cells. So the combination of oncomodulin plus a cyclic AMP analog is causing extensive axon regeneration. Conversely, in loss of function experiments, what we find is that if we induce inflammation in the eye by injuring the lens, or subsequently we use zymosan, there are other agents that will cause this massive inflammation without injuring the lens. But if we block oncomodulin with this peptide, the P1 peptide, we pretty much block most, if not all, of the regeneration. However, what we find is that oncomodulin by itself is not really sufficient, even with cyclic AMP, to cause regeneration. So if we go outside of these beads, what we find is even when we block oncomodulin, so this is looking at the survival of retinal ganglion cells. This is the normal survival in rats of retinal ganglion cells. With nerve injury, we lose about 75, 80% of the cells or more. But after lens injury or other inflammation, we've doubled the survival of ganglion cells, and blocking oncomodulin, either with the P1 peptide or with an antibody, a neutralizing antibody to oncomodulin, we still have this cell survival. So this is telling us that there must be complementary molecules present with inflammation that support the effects of oncomodulin. And in fact, when we only use recombinant oncomodulin without beads, we find that this is insufficient to cause regeneration. And in fact, what's happening is that just injecting the beads itself causes a little bit of inflammation, and that's enough to boost the effects of oncomodulin and cyclic AMP. So what else is associated with inflammation that enables this regeneration to occur? So going back a number of years, what Yu-Chin Yin found is that when we test multiple trophic factors in the presence of oncomodulin and cofactors, what we find is that only one trophic factor, SDF1 or CXCL12, is the only of these many trophic factors that we tested, BDNF, CNTF, GDNF, and so on. It's the only one that complements the effects of oncomodulin. And what we find is that SDF1 itself is heavily expressed in the infiltrative macrophages. So normally in a resting state, macrophages do not make much SDF1, but when we activate them by these pro-inflammatory stimuli, the F480 positive cells, that is, F480 is a stain for it's a surface antigen in mouse monocytes. We find that the monocytes, the macrophages, are expressing high levels of SDF1. So then when we test SDF1 in vivo, what we find is that SDF1 alone is enough to cause regeneration. This is not true of CNTF protein, but I'll show you in a minute. Gene therapy works. It's not true of BDNF. It's not true of a whole bunch of other trophic factors. And when we combine SDF1 with oncomodulin and cyclic AMP, we're pretty much mimicking the effects of zymosan in this case as an agent to produce intraocular inflammation. And then conversely, in a loss of function experiment, when we block these two proteins, we block oncomodulin with the P1 peptide, we block SDF1 with this other pharmacological inhibitor of the receptor that SDF1 uses, CXCL4, we completely eliminate regeneration. So this is telling us then that I'm skipping this, I'm sorry, I forgot. So here we've induced regeneration by a very powerful means that I'll get to in a minute. This comes from Solmara's work. But here, I'm jumping a little bit ahead of myself. We induce considerable regeneration by combining inflammation together with something that was discovered by my neighbor, Jigang He, here at Children's Hospital. That is a deletion of the P10 gene in retinal ganglion cells that also produces regeneration. If we combine the two, we get tremendous synergy. But if we add SDF1 to this combination, here is this very effective combination by itself, this gray bar here, looking at the number of regenerating axons at these various distances. But when we add additional SDF1, we have hundreds of axons that make it all the way to the end of the optic nerve. That's shown over here in this A2 panel. This is probably the most axons we've ever seen regenerating the full length of the optic nerve. And along with that, some of these axons are pretty clever, and they find their way into the optic tract and into the lateral geniculate nucleus, shown over here. And so the summary of this part of the talk is that intraocular inflammation, that can be done, you don't want to do this by lens injury, not very clinically applicable, but by using another pro-inflammatory agent. We have an infiltration of macrophages that make SDF1 and other molecules. They also make oncomodulin, but neutrophils make enormous amounts of this oncomodulin protein, and together this stimulates regeneration. And the part of this work that I think is really, the significant part of this work is that we can mimic these effects just using these defined molecules. So this may bring us closer to clinical translation. When we combine methods that overcome cell extrinsic suppressors of axon growth, so here we've injured the optic nerve alone, no treatment to stimulate regeneration. Here is one method to overcome cell extrinsic suppressors of growth, use of a dominant negative NOGO receptor. This is work we did with Jigang He, published back in 2004, or a way to inactivate the row A. Row A is a small GTPase that's part of the intracellular signaling machinery that shuts off axon growth in response to inhibitory molecule. The C3 protein, as shown by Lisa McKerracher, overcomes row A, but nevertheless, it doesn't give you much regeneration. However, when we combine these treatments to counteract cell extrinsic suppressors of growth, when we combine it with treatments to activate the intrinsic growth state, we get so much regeneration. Here, we've induced inflammation, and we've blocked signaling through row A. And here is our injury site. But histologically, we don't even see the injury site anymore. We get so much regeneration past the injury site that where the crush site is pretty much obscured. So remember now, this is an example of a CNS pathway in which you would normally have no regeneration whatsoever, and it's now possible to get lots and lots of regeneration, as Posadia knows as well, and some of the rest of you. And if we also go back to this paper by Martin Berry, we see something very interesting. There's a panel in his paper that he doesn't comment on. These are the experiments where he implanted these peripheral nerve grafts into the back of the eye. So R is where the retina is, G is the graft, and lo and behold, these peripheral nerve grafts are filled with inflammatory cells. And so this is probably the reason that Berry got extensive regeneration, and that is not because of molecules, not because of trophic factors made by the peripheral nerve grafts, but possibly because the grafts themselves are filled with inflammatory cells. And I've talked to Berry since then, and that's pretty much the way he now interprets his experiments. So, going the other direction, this direction. Okay, so this is, has a mind of its own. So this is the work of Takuji Kurimoto. We're looking at now the synergy between activating the intrinsic growth state of retinal ganglion cells by causing intraocular inflammation. It turns on all these genes associated with axon growth by virtue of expressing oncomodulin and SDF1 as trophic factors. We're getting lots and lots of regeneration, as shown in the second image here. What Jigang He's group discovered in 2008, 2009, is that deleting the P10 gene in retinal ganglion cells, this is a way of de-repressing signaling through the PI3 kinase AKT pathway. As described by Zhigang, this gives extensive regeneration by itself. But the question is, what happens if you put these two things together? Will it just be that, one occludes the other? It'll be about the same level of regeneration as either one alone, or maybe an additive effect. But the story is that it's an incredible synergy between these two effects. And so when we combine the two, we get extensive regeneration. So just 2 weeks after optic nerve injury, we already have some axons making it all the way down the length of the optic nerve. And so Silmara's work, which I hope you recall, this kind of historic paper by Silmara, looked at the effects of combining these pro-regenerative treatments over a longer period of time. So in Silmara's work, what we're showing up here, where's my arrow? Here is the injury site over here at the asterisk. And again, Silmara is getting so much regeneration distal to the injury site that you can barely see the injury site anymore. You're seeing loads of axons extending the full length of the optic nerve with enough time. Some of these very clever retinal ganglion cells are sending axons into the dorsolateral geniculate nucleus, superior colliculus, the nuclei of the accessory optic system. And what Silmara went on to show is that you can begin to see some recovery of genetically encoded behaviors, such as the optomotor response. So I wanted to talk very briefly about another trophic factor that people in the field have been very interested in, and that is CNTF, ciliary neurotrophic factor. So ciliary neurotrophic factor has gotten a lot of attention in ophthalmology. There are clinical trials in glaucoma and these other ophthalmic conditions that you see here, listed here, but many labs have also used it to produce optic nerve regeneration. But the interesting thing is that CNTF gene therapy, that is when you have an adeno-associated virus that's injected into the eye, CNTF gene therapy is in fact a very effective way of inducing regeneration, but the recombinant protein has almost no effect whatsoever, although what Zhigang He's laboratory showed is that if you delete SOX3, a repressor of signaling, through the pathway that CNTF signals through, the JAK-STAT pathway, then CNTF does have some effects. But the recombinant protein doesn't work. The gene therapy is enormously effective. So what accounts for this difference between the gene therapy and the use of the recombinant protein? So this is work by Lily Xie, just published in PNAS just a few months ago. And what Lily found, going back to the literature, this is a paper by my neighbor, Connie Sepko, the top one, and that is that any virus injection into the eye causes some level of ocular inflammation. So then that's a little bit of a hint of what's going on. But then this paper from Chi Tsui, an old friend, showed that CNTF happens to be a trophic factor for inflammatory cells. So if you inject the virus encoding CNTF, you are combining these two effects. You're causing some level of inflammation, and then CNTF amplifies that inflammation. So the hypothesis then is that CNTF gene therapy causes lots of regeneration because it's causing lots of inflammation. So to test this possibility, Lilly depleted neutrophils using an antibody to a neutrophil antigen, LY6C. It causes this very effective depletion of neutrophils. Here in our cell sorting pattern, here's where the neutrophils are. They're completely lost after this immune depletion of neutrophils. And what Lilly finds is that neutrophil depletion almost completely eliminates the effects of CNTF gene therapy. So what is it then from inflammation that's causing this regeneration? And so, of course, we tested our favorite candidates, oncomodulin and CXCL12, SDF1. And it turns out they are only mediating a little part of what CNTF gene therapy does. There's a whole other trophic factor, a whole other chemokine that's being activated by CNTF gene therapy. And that is the chemokine CCL5. So CCL5 is well known to be an activator of microglia, but it turns out it's also a potent trophic factor for retinal ganglion cells. So first looking for whether CNTF gene therapy causes elevation in CCL5. This, in fact, was reported by Shuei et al. In PLOS One among many chemokines that are elevated by CNTF treatment of the retina. So we replicate Shuei's finding that CNTF gene therapy causes this elevation. And then here, we're combining CNTF gene therapy with the anti-LY6G, where we've depleted neutrophils, and this elevation of CNTF goes away. Remarkably, even if we look in the blood, throughout the entire circulatory system, all the blood circulating blood neutrophils have elevated CCL5 because we've used CNTF gene therapy just in the eye. And we've actually seen this for STF1 as well. Just doing manipulations, just doing gene therapy in the eye changes the entire circulatory system, the inflammatory cell gene expression profile. So the complementary question is whether retinal ganglion cells can respond to CCL5, and indeed they express the receptor CCL5. It's called CCR5, receptor 5. Retinal ganglion cells are noted here by staining with RBPMS, an RNA binding protein, and the CCL5 is found on the cilia that extend from retinal ganglion cells. So we're learning now that the cilia that extend from cells can be rich in receptors, and receptors that are expressed on cilia are kind of in a privileged position for cell signaling because of their direct connection to the cytoskeleton. They can influence what's going on in the cell body very rapidly, and we're finding, and other people have found, other receptors on cilia that have very potent signaling. And so here, then, in a loss of function experiment, nope, let's see if, yes. So here we used CRISPR-Cas, where we combine, where the retinal ganglion cells are expressing Cas9, and now we're also expressing a virus, expressing a small guide RNA to the receptor, to CCR5. So this enables us to selectively remove the gene for the CCL5 receptor, for CCR5, only in retinal ganglion cells. So it's kind of a very surgical way of making retinal ganglion cells unresponsive. And what we find is that retinal ganglion cells are no longer responding to CNTF gene therapy. The top panel will be the positive control. We knock out the receptor in retinal ganglion cells, and they can no longer regenerate their axons. And then conversely, in gain-of-function experiments, recombinant CCL5 is more or less mimicking the effects of recombinant CNTF. So the upshot of this story is that the effects of CNTF gene therapy also involve neuroinflammation, but unlike just using lens injury or zymosan, whose effects are mediated through oncomodulin and CNTF, the effects here are mediated through CCL5 primarily, though there's some oncomodulin present. So the conclusion from this part of the story, and that's most of what I wanted to talk about today, is that at least 3 proteins associated with intraocular inflammation, oncomodulin, STF1, and CCL5, are potent growth factors for retinal ganglion cells. And interestingly, more or less all of these have been on nobody's map of trophic factors for retinal ganglion cells. You know, we think about BDNF, CNTF, which really don't have much effect by themselves. But these other trophic factors are really potent, when tested directly on retinal ganglion cells. So we're now kind of doing a broader survey of ligands to receptors on retinal ganglion cells to see if there are any more surprises waiting for us. Another example, whoops, that was premature. the work of Sherry Peterson, somehow on Zoom, the controls on PowerPoint don't work quite the same. So this is the work of Sherry Peterson, who's looking at another aspect of inflammation, and that is the inflammation that occurs in the optic nerve itself. So when we do our surgery, our injury to the optic nerve, we are getting an enormous infiltration of macrophages, of peripheral macrophages. They're stained here for the complement receptor, CR3 complement receptor. And what you're seeing here is that we're also staining in green for the myelin basic protein. And what we see is that the infiltrative cells are eating myelin. And it turns out this is one of the enabling factors that allows regeneration to take place, is that you need some positive stimulus to activate the intrinsic growth state of the neurons, but you also need the inflammation at the injury site to degrade the myelin, as well as suppress the formation of a scar at that injury site. And so what Sherry has shown is if we delete the proteins, if we delete the genes that activate the immune reaction to myelin, this involves the classical complement cascade. So the initiating step of the classical complement cascade is deposition of the C1q protein on damaged tissue. And what Sherry finds here is if we induce regeneration, I'll get to this method in a minute, but TPEN is a chelator of zinc. This is one method to induce regeneration, as shown here in the littermates. But when the C1q complement protein is deleted, we strongly diminish our regeneration. Ditto for regeneration induced by zymosan plus cyclic AMP, that's oncomodulin, SDF1. Here's the regeneration. If we don't have complement protein, we diminish the regeneration. And here is our super method for very strong regeneration. Oncomodulin, an shRNA to P10, elevation of cyclic AMP causes enormous amounts of regeneration, and that's partially diminished by blocking C1q, doesn't even attain statistical significance, and part of that's because this combination actually turns off the genes in retinal ganglion cells that cause retinal ganglion cells to avoid myelin. It actually shuts down expression of the NOGA receptor. But in any event, we find that the same is true if we delete the gene for C3, that is the kind of the node of the complement signaling pathway that more or less exactly phenocopies the effects of knocking down C1q. And likewise, if we delete the complement receptor, CR3, that's expressed on microglia and then on infiltrative monocytes that acquire the properties of microglia within the central nervous system, all of these are pretty much causing identical effects in suppressing the regeneration that we get by these three different methods. And then when Sherry looks at higher power, what we find is that the infiltrative monocytes, in fact, are indeed eating myelin. So this is why the injury itself actually is providing an environment that's both suppressive to growth by virtue of the scar that forms, but also in some ways enabling for growth by virtue of the infiltration of cells that are phagocytosing, the debris. And then what Nick Hanavis and Solmara did was kind of a gain-of-function experiment, where here we are injuring the optic nerve. We get no regeneration, of course, distal to the injury site. Here, we've induced regeneration using this combination of deleting the P10 gene, oncomodulin, elevating cyclic AMP. As expected, we're getting a lot of inflammation, but when Solmara and Nick cause a secondary injury that's causing lots and lots of inflammatory cells to invade here, we're getting enormous amounts of regeneration in this zone that's filled with inflammatory cells. So in a way, this is telling us, this is not an experiment you want to try at home, but this is telling us that if we elevate inflammation, if there's some way we could capture the positive effects of inflammation, we can greatly amplify the amount of regeneration that we get in this model system. Okay. So I don't have much time to talk about this. I just wanted to mention that, of course, inflammation has its negative side. And one place where this is very obvious is in the recent work that Shane Littleout did with the late Ben Barres, one of the greatest of all neuroscientists, sadly passed away 3 or 4 years ago. But this is part of a Nature paper published in 2017. This is an illustration that I found online, and that is that injury to the optic nerve is by virtue of activating microglia, and we know now also infiltrative cells, infiltrative monocytes, we get an elevation of these signaling molecules, interleukin-1 alpha, TNF alpha, and not a chemokine, but the C1 complementary protein. And what Littleout and Barres showed is that this causes astrocytes to become toxic. Astrocytes go into the so-called A1 state, a neurotoxic state, and in this state, they cause neural damage. So here in this, this loss of function experiment, remarkably, well, this could be done two ways. It could be done with antibodies to these three molecules, IL-1 alpha, TNF alpha, C1q complement. If you add the antibodies, this would be the normal loss of retinal ganglion cells 14 days after optic nerve crush. It's a little exaggerated, but what they're showing is that if you combine triple antibodies to these three proteins, you save the retinal ganglion cells, at least at 2 weeks. And here is a gene knockout experiment where they've knocked out the genes for interleukin-1 alpha, TNF alpha, C1q, and the normal loss of retinal ganglion cells that you would see. This looks like a more realistic number. Oh, this is a 7-day number of retinal ganglion cell loss. The ganglion cells are completely protected. So we're now doing further experiments with Littleout along these themes. So the model then is that optic nerve injury activates microglia, but also infiltrative cells. That causes the elevation of TNF alpha, interleukin-1 alpha, C1q, that polarizes astrocytes from the benevolent A2 state to the A1 state, causing the death of retinal ganglion cells. And then there are many papers, including a few from our lab, the ones that have the asterisks over here, that link TNF alpha to the retinal ganglion cells in glaucoma, papers that have come out long before we started working in this field. And this is from a paper that I was involved with Joan Miller, the chairman of ophthalmology at Harvard, Dimitrios Vavas, Min, or Irene Rowe was the first author of this paper. What we show is that if we experimentally induce glaucoma in a mouse model or in a rat model, two different papers use two different models, we get this large elevation of TNF alpha. If we use etanercept is a decoy receptor for TNF alpha. It's the external part of the TNF alpha receptor linked to an FC, the FC portion of IgG, to make a soluble receptor that just sort of swims around. And this etanercept, which is used clinically, and that was kind of the question of this study, is whether a clinical treatment could suppress the effects of glaucoma in an experimental model. So what we're seeing is that etanercept at high enough concentrations is greatly lowering the concentrations of TNF alpha circulating throughout the body. And normally then, the optic nerve head, we're looking here at the microglia marked by TNF alpha when they're activated. GFAP as a marker for activated astrocytes with optic nerve, with elevation of intraocular pressure, an experimental model of glaucoma, we see that there's a huge infiltration of microglia at the optic nerve head. This was also reported by Monica Vetter, also studied by Gareth Howells and Simon Johns, this infiltration in the experimental glaucoma model. These microglia are making enormous levels of TNF alpha, which is acting back on the microglia as a feed-forward mechanism, as an autocrine mechanism to keep the microglia activated. And along with this, the astrocytes go into an activated state where they elevate GFAP, and my guess is that they would be elevating other markers of the A1 astrocyte state. Whereas with etanercept, we block most of this. We block the infiltration of microglia at the optic nerve head, the elevation of TNF alpha, the activation of the astrocytes at the optic nerve head. And this is a little bit neuroprotective, but this is only with kind of minimal treatment with etanercept. And the idea is that perhaps blocking TNF alpha, if we sort of optimize the treatment, could become a clinically viable way to suppress retinal ganglion cell loss in glaucoma. I'm going to skip this next part about the presynaptic effects of blocking zinc, which accumulates in the terminals of amacrine cells after optic nerve injury. If we chelate zinc, that's enough to protect retinal ganglion cells. It's also enough to cause some regeneration. And then if we also do a combinatorial treatment here of combining everything with T-pen as a chelator of zinc, this is another way to get enormous amounts of optic nerve regeneration. And then the final thing I wanted to talk about is what are retinal ganglion cells thinking about when they regenerate their axons? And so this was a study using transcriptional analysis, looking at the gene expression profile of retinal ganglion cells when they are induced to regenerate their axons, compared with just injury alone, looking at the pattern of gene expression at different time points of regeneration, and then using bioinformatics to predict the transcription factors that are accounting for the changes in gene expression. So illustrating that here, we've induced regeneration by this strong treatment of oncomodulin, cyclic AMP, P10 deletion. So get lots and lots of regeneration, high levels of RGC survival. So the regeneration is shown in B. The survival of ganglion cells is doubled, as shown in C. And then we also did experiments with Jeff Goldberg at Stanford. Jeff, among the many things he's done, also did a study with Ben Barris, published in 2002, showing that retinal ganglion cells lose their intrinsic capacity for rapid axon growth that they normally have as they're developing their connections somewhere around postnatal day 1. By postnatal day 2, they no longer, when put in culture, can grow axons vigorously. So what we're hypothesizing here is that regeneration is the opposite of this developmental loss during development, and that the gene profile of regeneration should look like the gene profile of early stages of development when retinal ganglion cells are developing axons, and that transcription factors that enable ganglion cells to regenerate axons are the same transcription factors that suppress or that enable ganglion cells to grow axons developmentally, and then there are transcriptional changes that shut off that capacity. And so, from this work, we carried out these experiments, did optic nerve injury, either performed a strong pro-regenerative treatment or not, just injury alone with sham treatment, dissected the retinas out, either at day 1. We were particularly interested in the earliest stages of activating the regenerative program, but we looked at later time points, comparing the regenerative state with the controls. We carried out fluorescence-activated cell sorting to isolate the retinal ganglion cells from all the other cells of the retina, and then carried out bulk-seq or RNA sequencing to compare the transcriptional profiles of the regenerating retinal ganglion cells from the non-regenerating injured retinal ganglion cells, and applied bioinformatics with our wonderful colleagues at UCLA, the group of Dan Geschwind. So the early parts of this work were done with Giovanni Coppola, later with Riki Kawaguchi, and parts of this experiment were done with Jeff Goldberg, where we also looked at the transcriptional profiling of retinal ganglion cells in development. And parts of this experiment, the study, were done with Mike Sofroniev and Clifford Wolf. This is under review now, this paper. But the really exciting thing is that the transcription factor, REST or NRSF, two different names, it's a well-known transcription factor that's known to suppress the expression of neural genes in non-neuronal cell types. what we find is that rest is also active in neurons, where it suppresses the active growth state. And so if we remove rest, we can turn retinal ganglion cells into an active growth state. Conversely, when retinal ganglion cells are first forming connections developmentally, rest then becomes elevated and shuts off that ability. And that's what stops retinal ganglion cells from being able to grow axons after postnatal day one. Of course, there are other transcription factors involved as well. But from our bioinformatic analysis, rest comes out as the #1 candidate of controlling genes differentially between the regenerating state and the non-regenerating state. And if we also look at the development, comparing embryonic development with postnatal day 5, rest is not the #1 candidate, but it's very close to being the top candidate as regulating gene expression. So then the question is, what happens if we manipulate rest in vivo? And that'll be, I think, the last slides I'm showing. And so what we find in cell culture, if we knock out rest or express the dominant negative form of rest that binds to DNA but doesn't activate the growth program, that causes some growth. If we combine that with oncomodulin, we get a lot of growth in culture, probably more than we've ever seen. In vivo, it increases the survival of retinal ganglion cells. This isn't looking at regeneration, but, this is in culture. I'm sorry, that's the cell survival in culture. Rest is elevating survival. And in vivo, what we find is that just knocking down rest or expressing a dominant negative form of rest is enough to give us a fair amount of axon regeneration, and it fully captures the effects of P10 deletion and the whole treatment in increasing the survival of retinal ganglion cells after optic nerve injury. And when we combine rest with oncomodulin and cyclic AMP, what happens with that? We get as much regeneration as we get from deleting P10, which is probably the single most effective method to induce regeneration reported in the literature, and not shown here. If we combine dominant negative rest with P10 deletion, we get regeneration somewhere over here, just crazy amounts of regeneration. So this is then a very important transcriptional regulator, in this case, a transcriptional suppressor of the regenerative state. And then in other work with Jeff Goldberg and Dan Geschwin, we've also identified ELK1 as another important transcription factor that works together with rest to control the regenerative state. So with that, the first part of the talk about inflammatory mediators that enable regeneration to occur, oncomodulin, STF1, CCL5, that inflammation within the optic nerve also clears myelin debris, also enabling regeneration to occur. This third point I went over very quickly about presynaptic zinc, and then the final point about rest as a key upstream regulator of the regenerative state. And of course, where this is all going is to try to figure out ways to combine what we have discovered, what other labs have discovered, in order to be able to restore vision in a clinically relevant manner. I also wanted to quickly mention work from other labs that's relevant here. Jigong's group has shown IGF-1 and osteopontin as alternate ways of activating the growth state. Other ways of blocking the growth state or activating the growth state is blocking other cell-intrinsic suppressors. Several labs have worked with GSK3B, particularly Bill Snyder's group, and Andy Huberman group expressing a constitutively active form of REB. That's part of the PI3 kinase signaling pathway. Blocking receptors to cell extrinsic suppressors. I mentioned the NOVO components, but also PTP sigma is a receptor to parts of the scar that forms at an injury site. Increased physiological activity. Kai Yu has worked in this area, and Andy Huberman, who's at Stanford. And what else do we have? Chelating zinc, work from our lab. And then transcription factors, work from Jeff Goldberg's lab on KLF4 and 9, overexpression of KLF7, CMYC from Jigong, his laboratory, and now our work with rest, and I should add ELK1. So knockdown of rest or overexpression of ELK1. And so here are the people who've done this work, harnessing the innate immune system, primarily with Chin, but also Sherry Peterson, Lily Xie, Kim Wang, Fanny Feng, whose work I didn't have a chance to talk about today. Full-length regeneration, this is Solmara's work primarily, and Huya, shown on the right, has worked in this area as well. Non-cell autonomous control of the growth state, that is the upstream effects of inhibitory interneurons. This is the work of Eric Lee, Yiqing Li, who worked on this project with wonderful work for several years. Paul Rosenberg, my close colleague here at Children's, who is kind of the encyclopedia of neuroscience. Nick Hanovis has worked on part of this project. Kim Wang, and then this work about the master switches that regulate the regenerative state, primarily the work of Yu Chin, with help from Huya Gilbert. And with that, let me skip this and just give the running credits for people who've been involved in this work. And I think I've left people off. This is a little bit old. But with that, I thank you for your attention, and I'll be very happy to take questions.
Thank you.
Sorry I went over. It's not as bad as I usually am.
It's fine. It's very nice. Thank you, Larry. So let's get to the questions then. I think Rafael is the first person to put up a question here. Where is he? There.
Hi, Rafael.
Hi, Larry. Thank you so much for the wonderful question, as expected. I have one question, which is maybe too general, and maybe you guys have not done anything in the field, but I would like to know what you think about. My question is, what would be, or what do you think is, or has anyone done quantitative studies to know how much correlation there is between the regeneration, as you find, and retinal ganglion cell survival on the one hand, and axonal transport on the other hand?
Great questions. So survival has been, we study survival incidentally in the studies. It's something that we always measure as part of the regeneration. But there are several studies that clearly dissociate RGC survival from regeneration. So clearly survival is a precondition for regeneration to occur, but there are agents that are powerful promoters of RGC survival, but have no effect, and in some cases are suppressive to regeneration. So one clear example of that is BDNF, a paper from Adriana Di Polo a couple of years ago, showed that if we add BDNF together with intraocular inflammation, you get fabulous RGC survival and you eliminate axon regeneration. Work from, I'm forgetting who did it, showed that BCL2 overexpression also is enormously beneficial for RGC survival, but no regeneration. And then some of the most interesting work has just happened in the last few years. The work from Derek Welsby and Don Zak has shown that the DLK and LZK signaling pathways give you very high levels of RGC survival, but it blocks enabling regeneration by other means. However, Welsby just had a paper in PNAS showing blocking another kinase, a MAP4 kinase, gives very high survival, but it allows regeneration to take place if you have the building stimuli present. So The answer is that they are dissociable from each other. Oncomodulin is a great promoter of regeneration, but not of survival. Other treatments cause great regeneration and block survival, or some of them great survival, but they enable or they block regeneration. So it's a very complicated question. And it's really going to come down to understanding the inner workings of the cells. Really, what are the transduction pathways that lead to survival, and what are the signaling pathways that lead to regeneration? But I'd say we don't really have a good general principle. What we do know now on this question you just asked, there was an amazing paper that just appeared in Cell from a group at Mount Sinai Hospital in New York, showing that overexpression of CAM kinase 2 gives astonishing RGC survival after optic nerve injury and in a glaucoma model and with NMDA toxicity to retinal ganglion cells. It's almost unheard of, the level of survival that they report. And they also, in one model, in the NMDA toxicity model, look at visual function, pattern discrimination. So there must be axonal transport, right, if they're seeing visual function being retained. But I haven't read the paper carefully. I wish they had done the glaucoma model, which is, obviously clinically more relevant for preservation of visual function. But, this is now a new, exciting development in the field.
Yeah, that's actually my point. I wonder how much of what's in the literature actually applies to, let's say, an optic hypertension model.
Right. Right.
Other than the traditional optic crush.
Right. No, there's a lot now happening with the glaucoma model. So, for example, one sad story is that the blocking of the DLK-LZK pathway is fabulous for preservation in the crush model, but it doesn't apply to the glaucoma model. However, the work from Shane Littleau now with the blocking of these signaling molecules, C1q, TNF alpha, IL-1 alpha, that does apply to the glaucoma model for preserving retinal ganglion cells. So I don't recall how much of that is published, but I think what we're seeing is that the optic nerve injury model may be, the cell death mechanisms are not identical. And we know that optic nerve injury is causing both inflammation as well as blocking the DLK pathway. Glaucoma doesn't seem to be about the DLK pathway so much as about the inflammatory arm of retinal ganglion cell damage. So inflammation and astrocyte polarization, at least from the animal models that we have.
Okay, and the last quick question. Do you have any data on the ultrastructural responses in the regeneration models that you have been studying?
Yeah, so Silmara is now doing a lot of EM work. Really, she did it on the triple treatment to promote regeneration, looking at axons at the EM level. But Samara is also now looking at neurofibromatosis, which damages the optic axons and causes death of retinal ganglion cells. And Samara has some amazing findings in a mouse model of neurofibromatosis. So it's going on, but there's not much out there yet. Thank you for the questions.
Thank you. Thank you. Thank you for the talk and all the questions.
And Rafael. Now Rosalia has a question.
Yeah, please.
Where is she?
Hi, Larry. Thanks for the talk. It was wonderful, as usual. It's so nice to see everything together.
Obrigado. Obrigado. And I'm sorry I didn't have time to include your work. I'm very aware of it as alternate ways of preserving ganglion cells, promoting regeneration. Yes, and I'm sure everybody here is aware.
Yeah. My question in our system, and I wonder how stable are axons that reach the thalamus, for instance?
Right.
Do you follow them after a couple of weeks to see if they stay there?
No, we really haven't. We really haven't done exhaustive, or thorough time course studies. So we don't really have a good answer to that. I think the closest we're going to come to a longitudinal study now will be the work that Silmar is doing in the neurofibromatosis model, where we're asking the question whether the axonal pathology is there from the very beginning, or whether it sets in, and whether treatments that we're proposing are enable the axons to remain stably. But I would say that I'm not aware of anything in the field that's done that, except that this new paper from the Mount Sinai group has gone out to a year to look at least the anatomy, and I haven't read the paper carefully enough to know whether the functional studies were done at a year after injury. So this is something I think we all need to look at carefully. It's very, very exciting result.
Yeah, I'm asking this because we are trying to solve this problem, because we do have regeneration up to the superior colliculus, but in the follow-up, we see that some of these axons actually degenerate.
Yeah.
So my idea is, if we are recapitulating development, probably there is a lot of competition for synapses, and I keep thinking ways of increasing the activity of these new axons.
Yeah,
So
no,
they gain the competition, and we keep them there.
I think that's a very good point. But in addition, I think you need to consider ongoing inflammation.
Mm-hmm.
So, and I think the question is how much, to what extent does the inflammation resolve itself? So we know that the axon degeneration is going to engender and be influenced by inflammation. We know at the level of the retina and at the crush site is going to be persistent inflammation. In fact, someone in my group, Kimberly Wang, just did an experiment simply injecting AAV into the eye and found that microglial activation persists. You know, it's there for weeks after. We haven't seen how long it persists. And we haven't seen how long the inflammation caused by optic nerve injury persists. We've only gone out to about a month or so. But it is, quite plausible that inflammation could be attacking the axons, as well as the factors you're talking about, activity, competition. So I think these are now at the front edge of research in the field, these questions you're asking.
Thank you. Thank you again.
Thank you, Larry. Thank you, Rosalia. Now, Eduardo has a question.
Hi, Larry.
Hi.
Thank you very much for your talk.
Thank you.
I was wondering here, thinking about these species that naturally regenerate the retinal ganglion cells, like amphibians and fish.
Yeah.
What do you think is the main difference? Is that the inflammation that is slow, or they are more capable of recapitulating these transcription factors that we have in development?
One might think that having studied it for 40 years, I would know the answer to your question, but I don't. Let me think if I could say anything intelligent. Well, people have claimed that one thing we know from Dan Goldman's work, Dan Goldman in Michigan, is that suppressors of regeneration and promoters of regeneration, like KLF family members, come on very quickly in fish during regeneration. So Dan Goldman has done a lot in zebrafish, and he reported many years ago that with regeneration, there's an upregulation of KLF6 and KLF7. But why that happens, I don't know. And I don't know that this is a double negative sentence. who are triple negative. I don't know that they do not have the suppressive suppressive transcription factors like KLF4, KLF9, REST. So I think, that's one level of asking the question. But another level of asking the question is how much of the positive effect reflects positive effects of inflammation. Because, we're seeing that it's definitely a multicellular phenomenon. It's really not entirely intrinsic to the ganglion cells. So I don't think we really have a good answer to your question. Come back in 40 years. We'll take it up again.
Okay.
Thank you, Larry. Thank you, Eduardo. Now I have a question from YouTube from Silvana Lodge. She's asking, because she's in YouTube, she's not going to appear.
Okay, that's too bad.
She's asking if it is better to use anti-inflammatory agents than calcium channel blockers to lessen glaucoma signals. But this is not usual in the clinical practice.
Right, neither one is in clinical practice. There is work on calcium signaling, I know, in glaucoma. Actually, I am perplexed that with all the interest in inflammation, TNF alpha and other aspects of inflammation, that this really has not translated into clinical practice. I can't really speak for calcium channel blockers, how widely studied that is. But I've spoken to ophthalmologists, Harry Quigley, Len Levine, Jeff Goldberg. These people, the ones who are very academic are well aware of the inflammatory component, at least in experimental glaucoma, and the fact that blocking inflammation is more or less, very effective in blocking the progression of RGC loss. So I can't directly address your question about these two approaches, but I can also express perplexity that none of this has yet entered clinical practice.
Mm-hmm. Thank you, Silvana. And thank you, Larry. Then there's a question from Viviane Valença. Do you want to appear, Viviane? She thanks you for the amazing presentation and has some questions. Sorry if I lost this conclusion, but what do you think about the OCM effects? Is something dependent on immune cells respond to the OCM, or is it directly effect on RGCs?
Yeah. No, I'm sorry. We did a lot of work in cell culture on OCM. So OCM binds to a high affinity receptor on RGCs. The receptor has been very elusive to us. Right now, we're very close. We have the receptor. So it is a direct receptor-mediated phenomenon. But in vivo, the OCM, even with elevation of cyclic AMP in vivo, still needs an additional factor to preserve retinal ganglion cells. So this is where the SDF1 comes in. And what we believe now is that the effects of inflammation can now be bypassed. One can substitute oncomodulin, cyclic AMP, and SDF1, and that is mimicking the effects of inflammation.
Mm-hmm. She also was wondering if you investigated the room effects on microglia. Could this molecule regulate the microglia activation state?
That's also a very good question. We know, so I'll answer a different question. CCL5 definitely will act on microglia, and whether there is an additional contribution in vivo by, it's not just the CCL5 effect directly on RGCs, but CCL5 causing additional effects through the microglia is possible. We just know that if we block the receptor on retinal ganglion cells, we get no effect. So Lily Xie in our lab has done the experiments with the oncomodulin effect. And what we know, again, is mostly, we can give a very, a pretty complete answer about the effects of oncomodulin being direct. one is that it works on purified retinal ganglion cells. The other is, now that we've identified the receptor, Lilly has used CRISPR-Cas and has knocked out the receptor in retinal ganglion cells, and that will completely lose the effect of oncomodulin without losing the effect, say, of PTEN deletion as a positive control. However, it is possible that when we use oncomodulin in vivo, there is a side-on to that process where it is acting on other cells that are providing ancillary factors that are acting on RGCs. So this is a difficult question, whether effects that we see in vivo, although we can do very, what I think are very clean loss of function experiments, the gain of function experiments always have the possible additional factor that other cells are also being activated at the same time.
Thank you, Larry. A last question as to like the clinical practice of giving corticoids to the patients in acute phases, and how will this impact on the inflammatory helpful elements of axon growth, and if that has been tested?
Right, no, that's great. So the answer is that, of course, completely, toning down the inflammatory reaction is certainly likely to be beneficial, but you will also lose the positive factors, the trophic factors. So I think this is really now opening the question of how can we fine-tune the inflammatory reaction in order to, suppress the detrimental effects and to amplify the beneficial effects. I know that's a very general question, but I think this is a real challenge to anyone thinking about neural damage, where inflammation is an inevitable part of the response to damage. And can we get the beneficial part and suppress the negative parts? And very important area for the future.
Thank you so much, Larry. I think now we've come to an end of the questions, and it was great. Thank you so much for your time, for the great talk, and for the answering all these questions.
Great pleasure for me. Really a great pleasure. This is it's really wonderful to see all you people here, and I hope this happens in person someday in the not so distant future.
Yes. We'll have Silmara in the end of the month, right? So,
yeah.
So that will be good. And we'll have a whole month on these Mondays talking about visual system regeneration and plasticity with inflammatory elements. I'll send you our plan. Maybe you can come in sometime. Thank you so much.
Bye, Larry.
Bye, everybody.
Bye, Larry. Good to see you.
Thank you very much.
Bye-bye.
Great to see you.
Really great. Bye-bye.
Recording stopped.